All right, so quickly remind me, what was the whole point of the light reactions? Cool. Make NADPH and make ATP. And these are now going to get used in the dark reactions. What are we accomplishing? What are we using these to accomplish? Yeah, we're going to turn carbon dioxide into sugars. So let's call it sugars, some sort. And that's the goal. We call this carbon fixation. Carbon fixation. Cool. Why might a plant want to build some sugars? Yeah, where else is he going to get them from, right? So notice, you and I, can we produce our own sugars without taking any food in whatsoever? Well, for a brief time, but eventually if I keep not eating, I will eventually die, right? So, but plants, so what do they need to live? Well, they need sunlight, because for photosystem 1 and 2 to absorb some light energy. So, and they need water, and they need some carbon dioxide. And that's what they need. I mean, there's some other things, there's going to need some nitrogen sources and stuff like that, but as far as principle to get energy and to make sugars and stuff like that, those are the three things they need. Light, water, carbon dioxide. Some other nutrients and things like that, but this is some of the main things they need to grow. They make their own sugars. You and I, we can't make our own sugars without making them from something in our diet at some point along the way. So, but plants, they got it going. So here's your Calvin cycle, the dark reaction. So how many of you guys have seen the Calvin cycle in some other class? Calvin cycle, lovely, lovely, lovely times. The whole point of the Calvin cycle is to make sugars. These are the dark reactions. If you notice, we are going to use up some ATP, so in a couple of different places. So, and we're going to use up some NADPH. All the stuff we made in the light reactions gets used up here. So, and our goal is to make some sugars. Now, we might make those sugars for one of two reasons. Why might I need a sugar? Energy, which would be catabolism, or for structures and biosynthesis, which would be anabolism. That's the two fates. We're going to make some sugars either for catabolism or anabolism. Take your pick. So this is not necessarily the only place. So as what we saw with the light reactions, not necessarily the only place this plant might get ATP. A plant also might, generate some more ATP from catabolism of sugars it makes and stuff like that later. So, cool. If we look, Calvin cycle sucks. There's no easy way around that, guys. I'm just going to let you know, Calvin cycle sucks. So, but let's look at some big pictures. Big picture. My goal in the Calvin cycle is to turn look at the top there, 3 carbon dioxide molecules into glyceraldehyde 3-phosphate. That is my immediate goal of the Calvin cycle. And it's a really complex process that gets us there. If you look, kind of like oxaloacetate is what you combine with acetyl-CoA to get the citric acid cycle going. In this case, it's going to be ribulose 1,5-bisphosphate combining with CO2 to get this process going. Now, ribulose 1,5-bisphosphate, I think it's convenient to keep track of carbons. And so ribulose 1,5-bisphosphate has 5 carbons. When I mix that with CO2, how many carbons will I get? 6. So it turns out we're doing this, all the numbers in red show how many times we're doing this. So that's only making it more confusing apparently. So, but 1 ribulose bisphosphate combines with only 1 CO2 initially to make, so in this case, it initially makes a 6-carbon sugar that's not stable and splits. And it splits into 2 3-carbon 3-phosphoglycerate molecules. So initially, we're skipping the idea that it formed an unstable 6-carbon sugar because it immediately breaks down. So forming 2 3-carbon 3-phosphoglycerates. So we're going to do this process in triplicate, though. We are going to take 3 ribulose 1,5-bisphosphates. How many carbons is that? 15. We're going to mix it with 3 CO2s, 1 each, for a total of now how many carbons? 18. And the payoff is I get one little teeny tiny 3-carbon sugar. So how many carbons did we use up here? 18. And I produce what? A 3-carbon glyceraldehyde 3-phosphate. And that was my goal. The other 15 carbons are just used around the horn to regenerate 3 more molecules of ribulose 1,5-bisphosphate to keep the cycle going, if you will. So a lot of going on here. Most of it is just to regenerate ribulose 1,5-bisphosphate. Only 3 of my 18 carbons turn into glyceraldehyde 3-phosphate. And so that's why the net process is really just 3 CO2s there turning into glyceraldehyde 3-phosphate. All the rest of it is just involved in the regeneration of ribulose 1,5-bisphosphate. So if you notice, on the whole pathway, I only put one enzyme on there, and it is by far the most important enzyme. That is ribisco. Ribisco, depending on what source you look at, it is either the most abundant or one of the most abundant proteins on the planet, enzymes on the planet anyways. Because pretty much all plants do this, and there's lots of them, right? So you definitely want to know a few things about ribisco. We'll talk about his regulation upcoming and stuff like that. If you notice, every single structure in here you have seen before. And you had to know them before, which means probably not the worst thing in the world to rememorize them if you forgot them. But every single structure, 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, so on and so forth. Every one of those you've seen before. They were all part of glycolysis, as well as gluconeogenesis for that matter. So just keep that in mind. Once you make this glyceraldehyde 3-phosphate, which kind of was the point here, well, you can wait till another one is made, and if you combine 2 of them together, how big a sugar would you have? You'd have a 6-carbon sugar, so you can make hexose phosphates here. And then from the hexose phosphates, you got options. You can turn it into sucrose. So classically, we often show you how photosynthesis is used to make glucose, but that's not usually what plants are trying to do. Plants usually store it as fructose. So even though your textbook might show glucose or something like that somewhere along the way, sucrose. So it's typically what we're headed towards. They might also store some sugars as starch, big glucose polymer. So cellulose, why might a plant want to make some cellulose? Yeah, cell wall, structures. So, and then also some 5-carbon sugars as well. So the glyceraldehyde 3-phosphate could also be involved in another direction, which we talked about. You could just shuttle it off and do some glycolysis. If you do some glycolysis with glyceraldehyde 3-phosphate, where's it headed? Yeah, you're going to turn it to pyruvate and then head it off, put it into acetyl-CoA and head it off to the TCA cycle, citric acid cycle. Cool. So those are your fates at the end of the carbon cycle. Notice it's a lot of work and a lot of process to produce that one little tiny 3-carbon sugar. Sorry. So if you recall glycolysis, in glycolysis, when you split fructose 1,6-bisphosphate, you made dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate, in that case, isomerized into glyceraldehyde 3-phosphate. That way you had 2 of them. Okay. So if you go back a step, so notice it does both. So here, yeah, we got no laser anymore. Oh, because that's the light. There's also light on this thing. That's confusing. So glyceraldehyde 3-phosphate here actually isomerizes in between dihydroxyacetone phosphate. It doesn't really matter. We just say that we have 6 of those total. So and the reason it doesn't really matter is that one of them comes out of the cycle, and that's the one we're going to use to do some anabolism or catabolism with. The others just keep shuttling up through the cycle to make ribulose 5-phosphate and then to replenish our ribulose 1,5-bisphosphate.
Okay, so the dihydroxyacetone phosphate can be converted to ribulose 5-phosphate as well?
So no, it's actually going to proceed through the glyceraldehyde 3-phosphate. Okay. So the key is, though, is they're in equilibrium together, and the triose phosphate isomerase can kind of go back and forth, but eventually it's the glyceraldehyde 3-phosphate that will lead to the ribulose 1,5-bisphosphate. Okay. Cool, welcome. So big picture, by the way, net reaction. Glad we went back to this. 3 carbon dioxides used. What do we produce? 1 glyceraldehyde 3-phosphate. What did it cost us? 9 ATPs and 6 NADPHs. So what if I wanted to make, say, glucose or fructose? So there's 6 carbon sugars, you got to double it. So you'd have to make 2 glyceraldehyde 3-phosphates, so on and so forth. Cool?
What are the big red numbers for?
The big red numbers show how many of each molecule are doing it in the cycle. So notice, we're actually doing this whole cycle in triplicate, is the unfortunate part. Because if I do it once, I don't do a whole lot of anything. It's not until I do it 3 times that I actually get something out of it. So that's why I'm going to start with 3 ribulose 1,5-bisphosphates. So, and it's really just happening 1 at a time. But by the time I've gone through this 3 times round, at the end of those 3 cycles, I've produced 1 glyceraldehyde 3-phosphate that I can use for anabolism or catabolism somewhere else. But all the rest of the carbons are staying in the cycle to replenish the ribulose 1,5-bisphosphate.
So the cycle in triplicate then is worth 9 ATP.
Yes, the cycle in triplicate. Notice every time we do the cycle it uses up 1 CO2. And that's why we're doing it in triplicate here. We're using up 3 CO2s. And all 3 times total use up 9 ATP and use up 6 NADPH.
Okay, and NADPH is also worth 2.5 ATP, or is it worth?
We're not going to talk about what it's worth in terms of ATP because we're not going to shuttle NADPH off to an electron transport chain to make ATP, right? NADPH is there to build stuff.
Okay.
And so we're not really going to equate it to ATP, although we could, maybe try. So, but it's definitely a high energy. NADPH would be considered a high energy something or other.
Okay.
So, cool. So if you look, go figure, light activates photosynthesis. It's amazing. So light turns on ribisco. So activates fructose 1,6-bisphosphatase. So and phosphoribokinases, if I can say that properly. Light also inhibits phosphofructokinase and glucose 6 phosphate dehydrogenase. So if you look, why would it be a good idea if light's turning off, say, phosphofructokinase? So you don't do glycolysis. If you got light, then let's build sugars, not use them up. So I've got light. I can make energy in the light reactions, and I can use all that stuff to build sugars. I don't need to be using up, catabolizing my sugars. So on and so forth. So make the light activation and light inhibitions make sense to you. Also some indirect mechanisms. Turns out most of the enzymes work best around pH of 8. I can do this. Magnesium ions. Light increases the amount of magnesium ions, which the enzymes need. Cool, and I don't know too much, to be honest, about thioredoxin activation, but somehow this governs the activity of other enzymes, so it's involved in some giant, grand regulatory way. So if we look a little more closely at ribisco itself. So we said that ribisco is activated by light. It's actually the ribisco genes also that are activated by light. What does that mean? That the ribisco genes are activated.
You start getting transcribed through signal transcription.
Exactly. We're going to start doing transcription and translation and making more ribisco. So we're actually making more ribisco enzymes. Cool. Ribisco is also activated by increased pH and by the presence of magnesium. We said before, what's the ideal pH for most of the enzymes involved? pH of 8. So a pH increase would make sense. So also activated in a funky carbonylation reaction involving CO2. So the only reason to tell you that it involves CO2 is that it means that when I have high CO2 concentrations, ribisco's activity is higher, which kind of makes sense. CO2 is the reactant, and in this case, the reactant turns on ribisco. So it turns out ribisco's got some issues, though. Ribisco is kind of an annoying enzyme. It's really slow. And it has a competitor, and that competitor is oxygen. So ribisco can fix carbon, but it also has some oxygenase activity. And from what we know at this point, the oxygenase activity is just kind of along there, can't get rid of it, and it's a waste, as we'll see. Other point of regulation, fructose 1,6 bisphosphatase is activated at high pH in magnesium as well, just like Rubisco.